We talked about the phosphagen system fueling or synthesizing ATP for about 10 to 15 seconds, and then the substrates, creatine phosphate, is depleted and will stop ATP synthesis. So if our effort is longer than 15 seconds, we have to have a mechanism to produce ATP. And so the next system that will produce ATP is called glycolysis. And glycolysis is anaerobic, and the actual term does mean the breakdown of glucose. And that glucose comes from either circulating blood glucose in the bloodstream or from stored liver or muscle glycogen, which then is transferred into glucose or transformed. In the final product of this breakdown of glucose, there is a substance called pyruvate produced. And that pyruvate is either converted to lactic acid, or else pyruvate will be transported into the mitochondria and enter into the oxidative energy system, or for what's called the Krebs cycle, that is producing ATP in the presence of oxygen. So what happens to pyruvate in this process of glycolysis, or this breakdown of glucose, determines if ATP is going to be just produced anaerobically, which is the case when we convert pyruvate to lactic acid, or if it's going to be used for oxidative or energy production in the presence of oxygen. But glycolysis in itself, it can fuel exercise for up to 2 minutes. And it occurs, glycolysis itself occurs in the sarcoplasm, or the cytoplasm of the muscle cell. And it uses enzymes in this process. It's a long process, glycolysis. There's 10 steps. You don't have to know all the steps. What you do need to know is that it produces 3 ATP molecules if we start with glycogen, and 2 ATPs if we start out with glucose. And the reason for that is it has to use ATP in the very first steps to transform the glucose into a usable molecule to form the ATP. So there again, we produce 3 ATPs if we start with glycogen and 2 ATPs if we start with glucose. In this process, it's not just that ATP is produced, but hydrogen ions are also released, and they are transported to the mitochondria through a transporter which is called NAD. So the hydrogen ions that are released in this whole breakdown of glucose bind with NAD, and NAD then transfers them to the mitochondria to be used later in energy production through when in the presence of oxygen, or oxidative energy production. If there's not enough oxygen available in the mitochondria to accept the hydrogen ions for further oxidation, the pyruvate will be converted to lactate. And many of you think of lactic acid accumulation in your legs with high intensity effort, and which eventually causes excessive fatigue and the cessation of effort. And this is what happens in glycolysis if that pyruvate is converted to lactate. So glycolysis, the process itself is stimulated by strong muscular contractions, ADP, adenosine diphosphate, accumulating in the muscle, free phosphates, and those are all things that we see with the breakdown of creatine phosphate in the phosphagen system, as well as accumulation of ammonia, and there's a decrease in the pH in the muscle, which results in a more acidic environment. So all of that will stimulate glycolysis, the process to start. And it's inhibited also by a much lower pH. When we see more ATP in the muscle, more creatine phosphate, more citrate and free fatty acids. So, in other words, when there's other substrates available, it will inhibit glycolysis. And how fast the process of glycolysis occurs determines on the availability of enzymes that speed up the reactions in glycolysis. And the book discusses what's called the rate limiting step. That's the slowest enzymatic activity in this whole process, which is PFK. And so this enzyme really determines the rate of glycolysis. And this is just a diagram that shows, if you follow at the top, if we start with blood glucose, we have to use ATP initially to transform blood glucose to glucose 6-phosphate. And if we start with muscle glycogen can more easily be broken down to glucose 6-phosphate without having to use an ATP in the process to produce ATP. And so eventually we use another molecule of ATP to form what's called fructase 1,6-biphosphate, which breaks then down into two separate molecules, and then those are put through the process of glycolysis. And in this process, with these steps, you can see how a hydrogen ions, which is the H, binds with NAD, and then it's taken to the mitochondria for to be entered into what's called the electron transport chain, and that's part of our oxidative or aerobic energy production. But in this whole process, we're producing 3 ATP and 3 ATP if we start with glycogen, and 2 ATP if we start with glucose. But have to remember that the NADH that's transported to the oxygen is an important part of this, because that will be used to either enter the mitochondria for oxidative energy production, but if the mitochondria cannot accept the hydrogen ions, the NADH will take the hydrogen ions to where the pyruvate is, which is the very end product of glycolysis, and the pyruvate, with the addition of the hydrogen ions, will be transformed into lactate. And it's lactate or lactic acid production that results in the increased acidity of the muscle and fatigue and the eventual need to stop effort. So with the hydrogen ions that are produced and potentially going to the mitochondria, you can see how glycolysis is used to produce ATP to fuel exercise, but it also produces products to be used later on in the longer oxidative or aerobic energy production system. And so we talk about fast glycolysis and slow glycolysis. Fast glycolysis is when our end product pyruvate is converted to lactate, and slow glycolysis is when our end product pyruvate is transported into the mitochondria to be used for further ATP production in the oxidative system. So fast glycolysis, that occurs when we don't have enough oxygen available in the muscle, and it produces this lactate, which is eventually converted to lactic acid. We see significantly decreased pH, more acidic environment in the muscle, resulting in muscle fatigue. And this is then going to inhibit glycolysis or this production of ATP. It also may affect calcium binding with the troponin or cross bridges forming. The acidic environment can also affect the action of enzymes, the enzymes that are involved in glycolysis. And when we have a cessation of the glycolysis, there's decreased energy or ATP production for muscular contraction. But lactic acid kind of gets a bad reputation because we all feel lactic acid when we do short, high intensity effort, and we feel intense burning in our legs and fatigue in our legs, and we know it's lactic acid production. But what it is lactic acid production. Lactic acid is also a substrate. We can also use that to fuel energy production in slow twitch, type 1, and cardiac muscle. So it also is a substrate. When it's a problem is when we can't metabolize it fast enough and use it for energy, that it starts to accumulate and we have this very acidic environment. And so lactate can also be involved in producing glucose, which is the substrate that we use for in glycolysis. And it can be used, again, it's oxidized or used in the presence of oxygen for fuel for the muscle. The other thing it can do is lactate can be transported to the liver where to be used and produce glucose, which can be used as a substrate for further glycolysis. And when it's transported to the liver to form glucose, it's called the Cori cycle. And this is just a depiction of fast glycolysis, which converts pyruvate to lactate when not enough oxygen is available in the muscle. And then the lactate is transported to the liver and converted to the glucose. The glucose comes back and is used again in the process of fast glycolysis. Now, as we start to accumulate lactate, we can't use it, convert it to glucose fast enough. We can't use it as an energy source in the muscles. We start to accumulate it. And the blood lactate threshold is where we start to see a significant increase in lactate in the bloodstream. And it usually occurs at about 50 to 60% of a person's max capacity in an untrained individual. But trained individuals can sustain higher intensity effort before they start to accumulate lactate. So they're more efficient at using this lactate and ridding it from the muscle, using it for energy before they've reached their capacity to metabolize it. And so we see this increased lactate production as we see an increase in anaerobic metabolism. And lactic, your threshold is very much an indicator of athletic performance because VO2 max is the maximal amount of oxygen a person can use to produce ATP or energy. But if a person can function at a higher percentage of their percent max VO2, their lactate threshold is higher, and so they will be able to perform at higher intensities before they start to accumulate lactate. And so the lactate threshold is a indicator of when a person is starting to shift from aerobic to anaerobic energy production. Okay, so you will say that the person with it's not just VO2 max. Two people can have the same VO2 max, the same ability to consume oxygen, but the person who can maintain their aerobic metabolism at a higher intensity before they start to produce higher amounts of lactate will be able to outperform the other athlete because they can exercise at higher intensities without producing lactate, or they can metabolize it. And marathon runners, their ability to maintain those fast paces without accumulating lactate is a reflection of they have this very high lactate threshold. So they can maintain aerobic energy production before they start to accumulate a lot of lactates. It's very high level. And